converted into malonyl-CoA. So what happens here? Malonyl-CoA goes up. And guess what? What does malonyl-CoA do? It is a precursor, as we indicated, for fatty acid synthesis. It blocks CPT1. So if we have too much acetyl-CoA, this will lead to malonyl-CoA being produced. This blocks CPT1. What happens to the beta oxidation? Decreases. That's why it's hard to lose weight by losing fat. Why? Because notice here that you're trying to burn some fat. Acetyl-CoA goes up. The minute it goes up, blocks basically fat burning. As simple as this. That's why it's hard to lose weight. This is a slide you've seen last time when we said this is an example of beta oxidation of myristic C14 acid. We go through cycles. So we have the beta oxidation, the first reaction, dehydrogenation, hydration, which is mediated by an enzyme. The enzymes that I highlighted for you in red, you need to know and expect questions related to it. Enoyl-CoA hydratase from the name. This basically, it will be followed by dehydrogenation, which is oxidation, where NADH is also is produced from NAD. This is why if you, actually the reaction in the previous slide here is inverted. This is wrong. Where is it? Yeah. First FADH and then secondly NADH. This is the right order. Then we have the addition of acetyl-CoA and cleavage there. This is one cycle, and this cycle occurs based on the number of carbon in each fatty acids. So if we have C14, how many acetyl-CoA we will be producing? 7 acetyl-CoA. Why? Look at the last step. The last step will produce 2 acetyl-CoA. But the number of cycles, we said, C14 divided by 2 minus 1. So it will be 6 cycles. You've seen this many times. So you have the fatty acid length, the acetyl-CoA produced, and then beta oxidation. So do you expect, if I were to ask a question, how many cycles required for beta oxidation of certain fatty acids and so on? Energy-wise, I know I like to make it complicated here by making the factor as 1.5 for FADH2, 2.5 NADH. In the exam, we'll try to make it easy, 3 and 2, okay? If you look into the yield from every oxidation cycle, each oxidation cycle will produce 15 ATP, okay? So for palmitate C16, how many ATP we will produce? 1 FADH2, this is all basically will be from each cycle, around 15 ATP. If you have 16, we're talking about each cycle, so 15 times. How many cycles for C16, palmitate?
7.
Will be 7, right? 7 times 15 will give you 106. If you take into consideration the activation basically needed to activate the fatty acid, which is 2 ATP. This is how we do the math. Okay. The next 2 slides, I know it's going to be heavy, but I'm going to ask you just to remember key things. You can summarize it in 2 sentences. We know that most of the fatty acids are what? Even or odd number? Even. But we do have, to some extent, odd number. So what do we do with that last 1? We're not producing 2 acetyl-CoA as you've seen in the last step. We're producing here propionyl-CoA. What happens to the propionyl-CoA? Well, it undergoes different pathways. I don't want you to know, except that vitamin B12 is needed for conversion of propionyl-CoA into succinyl-CoA, which is an intermediate in the TCA cycle. This is what I need you to know from this whole thing, because mostly it is even, not odd. But vitamin B12, if we have vitamin B12 deficiency, there is a problem with fatty acid metabolism of odd number, and this could lead to accumulation of these odd number of fatty acids. What about unsaturated fatty acids? We already discussed this. Because of the time, I'll let you read it. I just needed to know one enzyme, which is the enoyl-CoA isomerase, and this would convert the cis double bond into trans, and then this would carry on with the process. So basically, you'll see the isomerase converting the cis into trans, so this would continue with the beta oxidation. This is for unsaturated fatty acids. That's all what I care about. The function of the enzyme, converting cis to trans to allow the unsaturated fatty acid to undergo beta oxidation. Are we clear? That's all what we need. Okay, ketogenesis, just quickly so we can finish on time. Ketogenesis, which we already know, we talked about it. Formation of ketone bodies. How does it happen? We said fatty acids. Oxidation is the source, where we have acetyl-CoA. 2-acetyl-CoA through an enzyme called thiolase gets converted into acetyl-CoA. Now a third acetyl-CoA, so 3-acetyl-CoA condense through HMG-CoA synthase. Beta-hydroxy, beta-methyl, glutaryl-CoA. HMG-CoA synthase will form HMG-CoA. We already know statins, right? You remember in cholesterol synthesis? So if you have HMG-CoA reductase, this will lead to cholesterol synthesis. This is where statins block. But if you have HMG-CoA ligase, here we have ketogenesis. So are we clear about the source of ketone bodies? Okay. HMG-CoA, we said HMG-CoA liase will convert the HMG-CoA into acetoacetate. Spontaneous decarboxylation will lead to acetone. And we have the beta-hydroxybutyrate dehydrogenase will produce beta-hydroxybutyrate. Which one out of these, what is known as ketone bodies, acetoacetate, beta-hydroxybutyrate, or acetone, is the most predominant one? Beta-hydroxybutyrate is the most predominant one. It's written right there. It's the most abundant one. Okay? Followed by acetoacetate. Now, why do you think people who have ketoacidosis, how do they smell? Fruity. Why? Acetone production right there. Okay? So in the liver, the whole idea is just to show you that in the liver we have the beta-hydroxybutyrate, acetoacetate. Now the acetoacetate with the succinyl-CoA will form the succinate acetyl-CoA TCA cycle. The liver cannot utilize ketone bodies, beta-hydroxybutyrate, acetoacetate. Why? Because it lacks an enzyme called beta-ketoacyl-CoA transferase. Another name you'll find in most of the textbooks includes succinyl-CoA 3-oxoacid-CoA transferase. Anyone expected, if I were to ask which enzyme, these are the same, the two names for the same enzyme, which is succinyl-CoA transferase, because it converts succinyl-CoA into succinate acetoacetyl-CoA through thiolase to acetyl-CoA TCA cycle. This is how brain, RBCs utilize ketone bodies in our body. In the presence of insulin, we do have enhanced glucose uptake by the tissues, and there's decreased mobilization of lipids, basically because we are inhibiting lipolysis. Whereas if there's absence of insulin or insulin resistance, there is decreased glucose uptake by tissues, and there's increased mobilization of lipids by adipocytes. This is the pathway which we said. Fatty acids, source of ketone bodies. Why? We said we have fatty acids, acetyl-CoA, but we said that because of during starvation, oxaloacetate is being pulled for gluconeogenesis. This pathway will be reduced, and we have the acetyl-CoA accumulation. This acetyl-CoA then goes into ketogenesis, and this is why we say the fatty acid is the source for ketone bodies. Are we clear? Ketoacidosis, I think you already had a lot of lectures about ketoacidosis, or you haven't? You already know about ketogenesis. I mean, for me, I already described it in terms of what do I expect from you. We have the only consequences of ketoacidosis. These are the causes of ketosis or ketoacidosis: diabetes, starvation, unbalanced diet, high fat, low carb diet, renal glucosuria. And here's another thing also. Since you're going for high fat, I know I'm done, but just wanted to comment on this. Why do we always go for high fat, low carb? Why do we have to take carb? If you're not taking carbohydrate, do you think you're going to be losing weight? From that figure, you should be able to tell me why. No, the answer and why. You're not going to lose weight if you don't take that low carb. Why? We need to prevent gluconeogenesis so that the TCA continues, so that we can burn more fat. Does it make sense why it's always high fat, low carb? You need that low carb, otherwise the TCA cycle would slow down. Okay, any question? It's the same slide, you just needed to know the enzyme, that's all. It's the same. This is acetoacetyl. Here we didn't talk about, I mean, the previous one, we have the ketone bodies. Now the ketone bodies in other tissues will be converted to acetyl-CoA in the brain and the RBCs. So this is where you start from. I mean, you have these acetoacetate with hydroxybutyrate, acetoacetate with hydroxybutyrate. Then in the liver, it doesn't happen. So the liver produces the ketone bodies but cannot utilize it. Other tissue could. If you have any question, please email it to me because I know that you have a session now. Okay, so just to give you a 5 minutes break before the second session. Thank you. Okay, sure. Just